Cancer is a complex disease. It's like a puzzle with many pieces. Each piece represents a different aspect of the disease. To solve the puzzle, we need to understand each piece. This is where precision diagnosis comes in. Traditional methods often treat cancer based on its location in the body. Lung cancer is treated as lung cancer regardless of its genetic makeup. But we now know that cancers with the same origin can have very different characteristics. These differences are driven by changes in genes, the blueprints of our cells. Imagine two patients, both diagnosed with lung cancer. They receive the same standard treatment. One patient responds well while the other doesn't. This difference in response could be due to underlying genetic differences in their tumors. Precision diagnosis aims to identify these differences. This allows doctors to tailor treatments for each patient, increasing the chances of success. Next-generation sequencing, or NGS, is revolutionizing cancer diagnosis. It's like having a powerful microscope that can read the entire genetic code of a tumor. This technology allows scientists to identify the specific genetic mutations driving a patient's cancer. NGS works by breaking down a tumor's DNA into smaller pieces. These pieces are then analyzed to determine the order of the building blocks of DNA, called bases. By comparing the sequence of a tumor's DNA to that of normal cells, scientists can pinpoint the exact mutations responsible for the cancer's growth. This information is crucial for developing targeted therapies. These therapies are designed to specifically attack cancer cells with those mutations. NGS is like having a detailed map of a city. It helps doctors navigate the complex landscape of a tumor and identify the best routes for treatment. Biomarkers are like signposts in the vast landscape of the human genome. They are molecules that indicate the presence of a specific disease, like cancer. In the context of NGS, biomarkers are often specific genetic mutations found within a tumor. These mutations can provide valuable information about the cancer's aggressiveness, its likelihood of spreading, and its potential response to different treatments. For instance, the presence of a mutation in a gene called EGFR can predict whether a patient with lung cancer will respond to certain targeted therapies. By identifying these biomarkers, doctors can make more informed decisions about a patient's treatment plan. They can choose therapies that are most likely to be effective, sparing patients from unnecessary treatments and side effects. The discovery and use of biomarkers are transforming cancer care. They are paving the way for more personalized and effective treatment strategies. Targeted therapies represent a new generation of cancer treatments. They are meticulously designed to specifically target cancer cells while leaving healthy cells unharmed. This is a significant shift from traditional chemotherapy, which can damage both cancerous and healthy cells, leading to substantial side effects. Targeted therapies work by interfering with specific molecules that are essential for the growth and survival of cancer cells. These molecules are often the products of genetic mutations identified through next-generation sequencing, or NGS. For example, if a patient's tumor has a mutation in the BRAF gene, a doctor might prescribe a BRAF inhibitor. These drugs are specifically designed to block the activity of the mutated BRAF protein, which is driving the growth of the cancer cells. The development of targeted therapies has been a major breakthrough in cancer treatment, offering new hope for patients with hard-to-treat cancers. The future of cancer treatment is indeed bright. Advances in next-generation sequencing and biomarker discovery are paving the way for more precise and effective treatments. We are moving away from the one-size-fits-all approach to cancer care. Imagine a future where cancer is diagnosed early, treatment is tailored to each patient's unique genetic makeup, and side effects are minimal. This future is within reach, thanks to the power of precision medicine. Studies have shown that patients who receive targeted therapies based on their tumor's genetic profile have better outcomes than those who receive standard treatments. For instance, in a study of patients with lung cancer, those who received a targeted therapy based on their EGFR mutation status lived longer than those who received standard chemotherapy. These successes are fueling further research and development in the field of precision oncology. The hope is that one day, cancer will no longer be a death sentence but a manageable disease.